Good afternoon. Today we are going to revise the last class and then we will learn about some applied aspects of the cardiac and the skeletal muscles. You know that the cardiac muscles, they are connected by the gap junctions, so they function as an electrical syncytium, and the ventricle function as a ventricular syncytium and the atria as atrial syncytium. This syncytium makes the contraction of the ventricle and the atria effective. Now let us revise about the excitation contraction coupling. When an impulse arrives in the presynaptic terminal, you know, a typical neuromuscular junction is not present in a cardiac muscle. Instead of it, when the nerve terminal, it comes near the muscle ending, it forms dilations, which are called varicosities, and they contain neurotransmitter vesicles. When an action potential arrives in the presynaptic terminal, this is easily transmitted by the gap junctions from the presynaptic membrane to the muscle end plate. This now travels along the sarcolemmal membrane down the T-tubule, and there are voltage-gated calcium channels which are present in the T-tubules. These channels, they open and there is influx of calcium inside the cell in the cardiac muscle. This calcium now helps in opening of the ryanodine receptor channel from which are present in the sarcoplasmic reticulum. So calcium influx causes release of calcium from the sarcoplasmic reticulum. This is known as calcium-induced calcium release. And the mechanism here is electrochemical coupling, because the electrical event causes rise in the calcium level and influx of calcium into the cell. So this is electrochemical coupling in the cardiac muscle. While in the skeletal muscle, it was electromechanical coupling. This calcium now binds with the troponin C, and the troponin-tropomyosin complex moves deeper into the groove of the actin filament, and the active sites, the actin filament, are now free. So myosin head, which has got ATPase activity and had stored the energy as ADP and PI, gets attached with the active sites of the actin filament. Then what happens? Then as soon as it attaches, there is the power stroke tilting of the head, and now ADP and PI are released. Again, another ATP binds with myosin head. As soon as ATP binds with myosin head, the myosin head loses its affinity for the active site of the actin filament and gets detached. Again, it breaks down ATP into ADP and PI and binds with the next active site of the actin filament. In this way, the myosin moves along the active sites of the actin filament, pulling the actin filament along with it, causing decrease in the length of the sarcomere. So, excitation followed by contraction of the muscle. Excitation, contraction coupling. This is also known as the sliding filament theory or ratchet theory. Until the level of calcium in the cell is decreased, this contraction will continue. For relaxation to occur, again the calcium needs to be pumped back by the sarca pump, that is the sarcoplasmic reticulum calcium ATPase pump, back into the sarcoplasmic reticulum or outside the cell by a form of secondary active transport, that is the sodium calcium exchanger. Then only the relaxation of the muscle will occur. Because the heart represents an electrical syncytium, With all the cardiac muscle cells contracting during a single beat, it is not possible to increase the force of contraction by recruiting more muscle cells. Moreover, tetany of the heart would be lethal because it would defeat the critical pumping action of the heart. You know, the heart has got the cardiac muscles, they have got long refractory period. So they cannot be either tetanized or fatigued. Tetanus is summation of contraction without any relaxation in between. So that is not possible in cardiac muscle because the refractory period is long. The long action potential found in the cardiac muscle which is due to the activation of the voltage gated L type of calcium channels results in the long refractory period, and this is the cause that prevents the tetany. Now, modulation of calcium influx through L type of calcium channels during an action potential can, however, provide the heart with a mechanism to alter cytosolic calcium and hence the force of contraction. A simple means of modulating the force of contraction of cardiac muscle in vitro is to vary extracellular calcium. You can see in this picture, the demonstration is given. Decreasing extracellular calcium from a normal range of 1 to 2 millimole to 0.5 millimole, for example, reduces the force of contraction. This reduction in force of contraction is not associated with the change in duration of the contraction because the kinetics of calcium sequestration by the sarcoplasmic reticulum and calcium extrusion has not been modified. We know that when there is sympathetic stimulation, you know, the force of contraction increases, the strength of contraction that is increased, the heart rate increases, as well as the relaxation period also decreases. So what does sympathetic stimulation cause? Let us revise this. Sympathetic stimulation, when it occurs during a flight and fight response. So there is activation of beta adrenergic receptors by either norepinephrine released from the nerve terminals, that is the sympathetic nerve terminals, or epinephrine released from the adrenal medulla into the bloodstream. You can see this picture. What has been the change due to beta adrenergic agonist? An increase in the force of contraction is termed as positive inotropic effect. There is also an increase in the rate of relaxation. So rate of relaxation increases accompanying the beta adrenergic stimulation that results in a shorter contraction. The increase in the rate of muscle relaxation is termed as positive lucitropy. The frequency of contraction of the heart also increases with beta adrenergic stimulation, and this is termed as positive chronotropic effect. So how does this epinephrine, norepinephrine act? Via the beta adrenergic receptors. They activate the beta adrenergic receptors in the cardiac muscle cells, which in turn activates adenylyl cyclase, increases CAMP, and thus promotes CAMP-dependent phosphorylation of numerous proteins present in the cardiac muscle cells. Both the voltage gated L-type calcium channels that are responsible for the calcium trigger and a protein associated with the Sarka pump called the phospholumban are phosphorylated by CAMP-dependent protein kinase. The combined action of these phosphorylation increases the amount of calcium in the sarcoplasmic reticulum. Specifically, phosphorylation of the sarcolemmal calcium channels results in more trigger calcium entering the cell, and phosphorylation of the phospholumban increases the activity of the sarca, thereby allowing the sarcoplasmic reticulum to accumulate more calcium before it is driven out of the cell by the sodium calcium antiporter. The net result is that sarcoplasmic reticulum releases more calcium into the cytosol during the next action potential, which promotes more actin-myosin interaction and hence increases the force of contraction. We have already learned about all these in the last class. So this was just a revision. Now let us see what is overdrive suppression. Overdrive suppression. You know, the heart beats at the rate of the AC node. There is autorhythmicity property of other cells also, AV node, AV bundle, then the Purkinje fibers, bundle of His. They also have autorhythmicity property, but the rate at which they can beat on their own is much less as compared to the AC node. The automaticity of pacemaker cells diminishes after these cells have been excited at higher frequency. This phenomena is called as overdrive suppression. Because the intrinsic rhythmicity of the AC node is greater than that of the other latent pacemaker cells in the heart, firing of the AC node tends to suppress the automaticity in other places. Overdrive suppression results from the activity of the membrane sodium-potassium ATPase. A certain amount of sodium enters the cardiac cell during each depolarization. The more frequently the cell is depolarized, the more sodium enters the cell per minute. At high excitation frequencies, the activity of sodium-potassium ATPase increases to extrude this large amount of sodium from the cells. The activity of sodium-potassium ATPase pump hyperpolarizes the cell because 3 sodium ions are extruded by the pump in exchange of 2 potassium ions that enter the cell. So more positive ions are moving out of the cell as compared to influx of positive ion into the cell. So the membrane becomes somewhat hyperpolarized. Therefore, slow diastolic depolarization requires more time to reach the firing threshold. When the overdrive suddenly ceases, the activity of sodium-potassium ATPase pump does not slow instantaneously or immediately, but temporarily remains overactive. This continued extrusion of sodium opposes the gradual depolarization of the pacemaker cells during phase 4, and it temporarily suppresses the cell's intrinsic automaticity. So this is about overdrive suppression. That is, the automaticity of pacemaker cells diminishes after these cells have been excited at a higher frequency. This phenomena is known as your overdrive suppression. Overdrive suppression. We have studied about the ryanodine receptor channels. Now we will see some abnormalities. You see here the ryanodine receptor channels. There is release of calcium from these channels. These channels are present in the lateral terminal cisterns of the sarcoplasmic reticulum. Now we will study about two clinical conditions. One is malignant hyperthermia, another is Brody's disease. Genetic diseases causing disturbances in calcium homeostasis in skeletal muscle include malignant hyperthermia and central cord disease and Brody's disease. So there are some genetic disturbances. This malignant hyperthermia is an autosomal dominant trait, but it has got life-threatening consequences. Anesthetics such as halothane or ether and muscle relaxants like succinylcholine can produce uncontrolled release of calcium from the sarcoplasmic reticulum, thereby resulting in skeletal muscle rigidity, tachycardia, hyperventilation, and hyperthermia. This condition is lethal if not treated. The drug of choice is dantrolene sodium. So until you do a muscle biopsy, you cannot detect this disease. So there is a mutation in these ryanodine receptor channels. You can see here the ryanodine receptor channels comparison in the skeletal and the cardiac muscle. So here in the malignant hyperthermia, there is some genetic disorder. So in the skeletal muscle, which are associated with the ryanodine receptor channel. So anesthesia with halothane, ether, muscle relaxant like succinylcholine. This causes malignant hyperthermia when these anesthetics are administered to these patients with the underlying disease. There results in malignant hyperthermia, and treatment of choice is dantrolene sodium. Now we come to Brody's disease. So what happens in this disease? This disease is characterized by painless muscle cramping and impaired muscle relaxation during exercise. Again, this is a genetic disease. While running upstairs, for example, muscles may stiffen and temporarily cannot be used. This relaxation abnormality is seen in muscles of legs, arms, eyelid, and the response is worsened in the cold weathers. This BD can be either autosomal recessive or autosomal dominant and may involve mutations in up to 3 genes. It appears that the BD results from decreased activity of the SARCA1 calcium pump found in fast twitch skeletal muscle. The decreased activity of SARCA1 has been associated with the mutation in the SARCA1 gene. Although there may be accessory factors that contribute to the decreased sarcoplasmic reticulum calcium uptake in the fast twitch skeletal muscles of the persons having BD disease. So this is associated with the abnormality of the Sarka pump, while the malignant hyperthermia, we have seen it is associated with abnormality of the ryanodine receptor channel. You can see the ryanodine receptor channel. Now we will see something about the rigor mortis. You know, for contraction, we require ATP. Again, for relaxation, we require ATP. Relaxation in the muscle, that is skeletal muscle, cardiac muscle, these are active processes. For detachment of myosin head, we require ATP. And for the calcium to be pumped back either into the sarcoplasmic reticulum or out of the cell, we require again ATP. So after death, what happens? The body becomes stiff, and because there is no more ATP, so the muscles go into rigor. There is no more relaxation of the muscle, because for relaxation, what you require? You require energy. There is no more energy available now, so there is stiffness of the body that develops. With the body composition, the temperature, and other associated factors, the time of this rigor mortis can vary until the degrading enzymes, they come into play, and finally the muscle relaxes. We can detect the time of death by seeing a body after death, say a post-mortem changes. If the body is warm, flaccid, the person is dead less than 3 hours. If the body feels warm but stiff, dead for 3 to 8 hours. This can vary with temperature, body composition, but somewhat like this. If body feels cold and stiff, dead over 8 to 36 hours. If the body feels cold and flaccid, that is dead more than 36 hours. Now we come to the dystrophin glycoprotein complex. We have already discussed about this. This is the, you can see the large dystrophin protein, which is a rod-like structure, which connects the thin actin filament to the transmembrane protein, the beta dystroglycan, in the sarcolemma by small proteins in the cytoplasm. These are syntrophins. Beta dystroglycan is connected to merosin in the extracellular matrix by alpha dystroglycan. The dystroglycans are in turn associated with a complex 4 transmembrane glycoproteins, alpha, beta, gamma, delta, sarcoglycan. This dystrophin glycoprotein complex adds strength to the muscle by providing a scaffolding for the fibrils and connecting them to the extracellular environment. Now we will come about some abnormalities with this protein, that is muscular dystrophy. 2 types we will discuss. One is the Duchenne muscular dystrophy, another is the Becker muscular dystrophy. So the dystrophin gene is one of the largest in the body, and mutations can occur at various sites in it. In Duchenne's muscular dystrophy is serious form of dystrophy in which the dystrophin protein is absent from muscle. So here the dystrophin protein is absent from the muscle. It is X-linked and is usually fatal by the age of 30. So this is a serious condition, the Duchenne's muscular dystrophy. It is X-linked and usually fatal by the age of 30. Now we come to the milder form of the disease, that is the Becker muscular dystrophy. Dystrophin is present but altered or reduced in amount. So in Duchenne's muscular dystrophy, dystrophin is absent. While what happens in Becker, it is altered or reduced in amount. Limb-girdle muscular dystrophies of various types are associated with mutations of genes coding for the sarcoglycans or other components of the dystrophin glycoprotein complex. So, in these persons, what happens? Gower signs becomes positive. When you sit down, you can easily stand up. But when the boy you see out here is trying to stand up, it is like that he is trying to climb up on his leg. This is climbing test positive or the Gower sign positive. This is typical of muscular dystrophy. Pseudohypertrophy of muscle occurs because now the scaffolding of the fibrils and the connection of them with the extracellular environment is lost or hampered. So this results in pseudohypertrophy of the muscle. You can see from when he's trying to stand, it is so difficult for the boy to stand. He is trying to as if climb up on his legs, climbing type of sign positive, that is Gower sign positive. This is the dystrophin glycoprotein complex. Now we come to the anticholinesterases. Anticholinesterases are agents which inhibit acetylcholinesterase. Let us revise about the neuromuscular junction. We have seen in the neuromuscular junction in the skeletal muscle, in the presynaptic terminal, there are acetylcholine vesicles. When an action potential arrives, what happens? There is change in the membrane voltage. This causes influx of calcium. This calcium is sensed by a protein called the synaptotapmines. So this creates a microdomain and helps in further interaction of different proteins. There are some proteins present in the vesicle which are known as V-snare protein, synaptobremin, and in the membrane there is T-snare protein. There are also RAB protein, MUNC protein, SNAP protein. So there is interaction between these proteins. As a result, the zipper opens and neurotransmitter is released like a zipper opening, released from the synaptic vesicle into the synaptic cleft. There may be total release of neurotransmitter or a phenomena called kiss and run. This neurotransmitter now is acetylcholine in this instance. So this acts on the acetylcholine gated receptor channels present on the muscle end plate. And it binds with the 2 alpha subunits, which causes opening of the channel, and there is influx of sodium and as well as there is influx of potassium, but influx of sodium is more as compared to influx of potassium because inside the cell is negative. This helps sodium to move inside while it prevents potassium to move out of the cell. And this way an action potential is generated in the muscle end plate. Now what happens? What is the fate of this acetylcholine? This acetylcholine is broken down by acetylcholinesterase into a choline and acetate, and choline is reuptaken by a clathrin-mediated transport for resynthesis in the presynaptic terminal. Now these anticholinesterases are agents which inhibit the anticholinesterase and prevent acetylcholinesterase from hydrolysis of the acetylcholine. You have seen use of this drug in myasthenia gravis. The drug was neostigmine. The drug was neostigmine. Now, this is the classification of acetylcholine esterases. Some are reversible and some are irreversible. Reversible are carbamates, physostigmine, neostigmine, pyridostigmine, edrophonium. These are the drugs. Then there are the acridine. Then the irreversibles are the organophosphates, diflos, ichtiophosphate, malathion, parathion, tebune, serine, soman. These tebune, serine, soman, these are nerve gases. Organophosphorus poisoning, you will see very commonly. Insecticides are taken sometimes for committing suicide in patients. You will be seeing them as you go to clinical classes. So, Baygon spray, you have learned about it. It is a carbamate. You have seen as a killer of mosquito, we use it. So, these are anticholine esterases. Anticholine esterases, reversible, irreversible. So, this is the classification. Now, we come to the long QT interval. So, there may be some abnormalities which you can see in this picture. This is the normal QT interval, and when there is some abnormality in the QT interval and the QT interval becomes long, that is called long QT interval. We have studied about this long QT interval in the last class. I am just demonstrating the picture in ECG that you will find in the long QT interval. Now, let us again revise about the digitalis. What does digitalis do? In a heart failure patient, you give digitalis. So, what does this digitalis do? This digitalis, it blocks the sodium, potassium, ATPase pump. So, the sodium, potassium, ATPase pump is the primary active transport. It provides the electrical as well as the concentration gradient required for running the sodium calcium exchanger. So now, as the sodium potassium ATPase pump has been stopped, the sodium calcium exchanger no longer can act. So what happens? Calcium accumulates inside the cell or inside the sarcoplasmic reticulum. So more calcium is available in the cell, which increases the force of contraction, that is ionotropic effect in a failing heart. Here you can see in this picture what happens. There is dilation of the heart. So dilated cardiomyopathy. You can see dilated cardiomyopathies in different conditions. We will later revise them again. So we will see dilated cardiac myopathy, what happens in them. Now just revise what is muscle hypertrophy and muscle hyperplasia. You can see here what is happening. The sarcomere is the fascicle. You can see the fascicle. What is happening? The sarcomere length is increasing. Here, what is happening? More and more myofibrils are added in case of hyperplasia. So, hyperplasia and hypertrophy both occurs in exercise. Persons who do exercise regularly, due to exercise, there may be hyperplasia or hypertrophy of muscles. In hypertrophy of muscle, there is increase in the length of the sarcomere. In the hyperplasia, what will happen? There will be addition of, what will be their addition? There will be addition of more myofibrils. Now let's come to the cardiac muscle hypertrophy. Now exercise such as endurance running can increase the size of the heart as a result of hypertrophy of individual cardiac muscle cells. Concomitant with this enlarged athlete's heart is improved cardiac performance as assessed by an increase in stroke volume, increased oxygen consumption, and preserved relaxation. Athlete's heart represents an example of physiological hypertrophy with beneficial contractile effects. In contrast, if exposed to chronic pressure overload, the heart may undergo either concentric left ventricular hypertrophy or dilated left ventricular hypertrophy with impaired functional consequences. Concentric hypertrophy is characterized by thickening of the left ventricular wall and represents a compensatory hypertrophy to the increased load. Dilated hypertrophy is characterized by increased ventricular volume, that is end diastolic volume. Both concentric compensatory left ventricular hypertrophy and dilated left ventricular hypertrophy have been shown to exhibit decreased contractile response to beta adrenergic stimulation. That is, there is decrease in the cardiac reserve, that is the contractile reserve. In dilated left ventricular hypertrophy, normal contractile function along with the Frank-Starling response may also be impaired. So what is Frank-Starling's law? Greater the initial length of muscle fiber, greater will be the force of contraction within physiological limits. So that is hampered now. The cellular and molecular mechanisms underlying the development of cardiac hypertrophy, however, remain still unclear, although there is an elevation in intracellular calcium, and this may be the cause. The link or links between cardiac hypertrophy, decreased cardiac performance, and impaired beta adrenergic response during chronic pressure overload still remains unclear. Decreased cardiac performance has been attributed to dysregulation of intracellular calcium. There is dysregulation of intracellular calcium, alterations in the level activity and phosphorylation status of variety of proteins, including L-type calcium channels, phospholamban, SARCA, ryanodine receptor channels, have all been implicated in calcium dysregulation, which is associated with a failing heart. The impaired beta adrenergic response of cardiac muscle after chronic pressure overload involves, at least in part, a decrease in beta adrenergic receptors because of internalization. There is internalization of the beta adrenergic receptors. A modest elevation in intracellular calcium as a result of increased contractile activity, for example, has been proposed to activate a calcium calmodulin dependent protein phosphatase, calcium calmodulin dependent protein phosphatase, which is calcineurin, that can dephosphorylate the transcription factor NFAT. NFAT, that is nuclear factor of activated T cells, and thus facilitating translocation of NFAT to the nucleus and promoting protein synthesis and hypertrophy. Activation of calcium calmodulin dependent protein kinase has also been seen in activation of transcription factor MEF2, myocyte enhancer factor 2, by promoting the dissociation of an inhibitor of MFE2. So these are the causes, different causes, and a brief discussion about the cardiac muscle hypertrophy. Cardiac muscle hypertrophy. So here we have learned about different cardiac muscle hypertrophy. Now we will discuss about familial cardiomyopathic hypertrophy, which occurs in approximately 0.2% of general population, but is a leading cause of sudden death in a otherwise healthy adult. It has been linked to genetic defects in a variety of proteins in the cardiac sarcomere, including myosin, troponin, tropomyosin, and myosin binding protein C, a structural protein located in the middle of the A band of the sarcomere. Familial cardiomyopathic hypertrophy is an autosomal dominant disease. This is an autosomal dominant disease. So we have learned about some types of cardiac myopathies: cardiac hypertrophy, muscle fiber hypertrophy, hyperplasia. We have learned about them. We have learned about the drug digitalis. We have seen the images of normal QT interval and the long QT interval. We have seen classification of the anticholinesterases. We have seen how the cardiac muscle, they function as a syncytium and which causes the cardiac muscle to contract effectively. We have learned about the excitation contraction coupling of the cardiac muscle, the skeletal muscle. We have learned about rigor mortis. Because there is no ATP available, what happens? The muscle remains in a state of contraction until degrading enzymes, they degrade the muscle. After that, the muscle becomes flaccid. So you can detect the time of death. Then we have learned about the pseudohypertrophic muscular dystrophy, the Duchenne muscular dystrophy, in which we have seen that the dystrophin protein is absent in the muscle, while in Becker, dystrophin is present, but it is either altered or diminished in amount. So we have learned about all these. Go back and revise this, and we will see on the next day, we will see about the smooth muscle. Thank you. See you. Bye.